Yeah, I apologize for the delay and I apologize for the inconvenience as well. Okay. So now we'll start with where we left off. In the last class, we know that we stopped till incomplete dominance, and the next part of non-Mendelian genetics with respect to inheritance of one gene would be codominance. So codominant condition, the example for codominant condition, what we are going to take is human blood grouping. Okay. So before we talk about the example and how it works, so the definition for codominance is whenever we have two alleles, what happens with these two alleles is both of them will equally dominate and they create a combined phenotypic effect. So in the last class, I told you very clearly what's the difference between codominance and incomplete dominance. So if you remember, what did I tell you? In incomplete dominance, what will happen? If A is one allele, B is another allele, both of them will combine to give rise to a phenotype called as C. Okay, that's what happens. No, if there is A is one allele and then B is another allele, if both of them come together, okay, they will give rise to a phenotype called as C. So that's what happened in your which one 4 o'clock plant or the dog flower, etc. So there was red flower, there was white flower, both of them gave mixed together to give rise to a pink flower. So that means a new phenotype was born in incomplete dominance. Whereas in codominance, what will happen? In codominance condition, we have a combined phenotypic effect. So that, for example, what will happen? The example is human blood group. In human blood group, we have three types of blood group that we already know. One is A blood group, B blood group, and you have O blood group as well as AB blood group. Sorry, four types of blood groups are there. Okay. Now, what are these blood groups? Blood groups are a particular characteristics. It's a very important characteristics in our physiology. So based on the presence of an antigen, okay, we can differentiate individuals into what kind of a blood group they belong to. Why it is necessary? Because we know that for us to two reasons are there. One is genetic reasons, to understand that there is so much of variability even at the at the cellular level. We have so much variation at the cellular level. That is the first condition. Second condition is we cannot randomly donate the blood to anybody, so enabling us to understand that there is compatibility for blood donation. That means only this particular blood groups can donate blood group to certain groups, or certain groups cannot donate the blood group to other groups. So this condition was very, very important to save lives. Okay, what is happening here? Let's see here. So if you see what is ABO, ABO is actually, these are the alleles which are located on the chromosome 9. Okay, we have our chromosome 9, right? On the chromosome 9, we have these alleles. Okay. Now, what are the alleles responsible? We have IA, we have IB, and then we have small I. So these are the three alleles, IA, IB, and small I are the three alleles which are responsible for us to give rise to the blood typing or the blood groups. Okay. So you see here in many conditions, like the past conditions that we saw, in a monohybrid cross or in incomplete dominance, you could see only two alleles, either it is capital R or small r, or it is capital T or small t. But in blood grouping, what you're seeing is there is IA one type of allele, IB as another type of allele, and then you have small I as another type of allele. So what does it suggest? It suggests that more than two alleles are present for a particular phenotype. So this condition is also called as a multiple allelic condition. Okay, there are multiple alleles for this condition. Okay, let's look at this. So this is your chromosome 9. So at the location, loci of a chromosome 9 is called as 9q34. So anyways, this particular thing is not important for our memory. But anyways, what we can understand that it is present on the 9th chromosome at this particular position. Okay. So what do we have here? We have the blood groups. What are the blood groups? We have A blood group, we have B blood group, we have AB blood group, and then we have O blood group. Okay. So what is happening? How do we tell that we have a person is having A blood group, B blood group, AB or O blood group? Now we have some solutions called as anti-sera. Okay. If I have, okay, A anti-sera and I put it to a particular group, if it clogs, okay, we call it as deglutination reaction, or if the blood clots together, okay, it becomes small clot kind of a reaction, then it is called as A blood group. A anti-sera reacts with A blood group, B anti-sera reacts with B blood group. Okay, for AB blood group, it can either react with A anti-sera or B anti-sera. It reacts with both of them. whereas the O blood group will not react with any of the antisera. So what are we talking about? How do I recognize that? Okay, antisera is something that we can use to recognize what blood group a person is having. What will I do? I have antisera A and then I have antisera B. Okay, these are the two types of antisera, isn't it? So now what will happen? A antisera A blood group. I will write it as BP. Okay, BG blood group. Got it? And then B antisera means it will react with B blood group. Got it? B blood group. And then I have AB blood group. In AB blood group, okay, what do I have? It reacts with both A and B antisera. Okay, antisera. So this is how I can physically examine which person is having which blood group. Whereas if I talk about O blood group, okay, O blood group, O blood group will not react with any antisera. What is the reason? Now, what are we talking about? Now, if I have a particular blood group, whether it is A or B or AB, now in these groups, what will happen? Because of the presence of IA gene or IB gene or both IA and IB gene, here a particular polysaccharides get synthesized on the plasma membrane of the red blood cells. Okay, we have the plasma membrane for the red blood cells, right? So a type of sugar, a polysaccharide starts producing on the particular blood group. Okay, if a type of a polysaccharide is synthesized, that belongs to the A blood group. If a B type of polysaccharide is synthesized, it is called as B blood group. If both the type of polysaccharides are produced, it is called as AB blood group. If there is no polysaccharide present, it is called as a O blood group. Now the question is, we know that in general forms, what is an antigen and what is an antibody? Okay, so we know that antigen is something of foreign nature, correct? No, something that does not belong to our body is called as what? It is called as an antigen. Do you agree with me? Excuse me. See here, what do I tell? What is an antigen? Antigen. What is an antigen? Antigen is something that does not belong to my body. That is called as an antigen. But if I say that I have A blood group, then what kind of an antigen I have? I have A antigen. If I have A blood group, then I have what antigen? A antigen. If I have B blood group, then what kind of antigen I have? I have B antigen. If I have AB blood group, then I have what kind of an antigen? I have AB kind of an antigen. So if something that does not belong to my body is called as an antigen, then how come if I have A blood group, I have A antigen? So the question is this. So to answer in a very simple term, I have A blood group. I cannot donate blood to a B blood group. Correct? No. If I have A blood group, I cannot give my blood to my B blood group guy. So for a B blood group guy, my blood is very foreign. So his blood will start reacting with my body. That's why it is called as antigen. Did you understand the concept? Okay, don't get confused because this is a very important, very sensitive question that you can come across in the NEET. So here you can see if a blood group is A, what kind of an antigen is there? See, in blood group A, I have antigen A. If a blood group B, I have antigen B. If a blood group is AB, I have antigen AB. If a blood group is O, I don't have any antigens. You get my point? So now, okay, what is an antigen? That's what. So Vicky is asking us, what is an antigen? So in a simple term, that's what I'm telling. Antigen can be anything. It can be a dust, it can be a pollen grain, it can be chalk dust, it can be smoke, it can be anything like bacteria, virus, any pathogen or anything living or non-living which does not belong to your body. If it enters, that is called as an antigen. Okay, that is a definition for an antigen. Okay, whereas in blood grouping, what happens is we get a little bit confused between the term antigen. So just that's what I was telling Vicky. If I have A blood group, if my blood group is A, let's say that I have A blood group, then I say that I have A antigen. Okay, why do I say that I have A antigen? Even though it's my own blood and my own blood has A type of polysaccharide, then why do I call it as A antigen? Because I, being an A blood group person, I cannot donate blood to a B blood group person. Correct, right? So if I donate my blood to a B blood group person, what will happen? The B blood group, the B blood will start attacking the A cells and then the clot starts forming in your blood vessels. So that is why we call this A blood groups antigen as A antigen, B blood groups antigen as B antigen, AB blood groups antigen as AB antigen. Did you guys understand this concept? Yeah. So in simple terms, if I have that particular blood group, then I will have that particular antigen. As simple as that. Okay, na? Okay. So this is one thing that you have to understand. And the second part of this is we understood what is an antigen and what kind of an antigen is present on that specific blood group that we understood. Now let us understand what about the antibodies. What is an antibody? Antibody is a protein. Basically, it's a protein that is meant to fight an antigen. Okay, it is your own. Your body produces antibodies as a defense system. It's like a defense system. It's like a molecular defense system. So what does it do? It produces antibody to fight against antigens. You get my point? So now what will happen? I am a A blood group person. I am a A blood group person. For me, which blood group is foreign, which blood group is away, or which blood group is not recognizable? Something away from A. Okay, something which is not A is always foreign for me. Okay, so now listen here. If I have a A blood group person and then I give the blood group B, then my body starts fighting the B blood group. In order to fight the B blood group, then what should I have? I should have a defense. What is my defense? Anti-B. Anti-B antibody will be there in my plasma. That is why my body will not accept the B blood group being a A blood group person. Did you understand? So in simple terms, what will happen, guys? If I have an A blood group, if a person's blood group is A, he will have what kind of an antibody? He will have B antibody. If a person's blood group is B, then what kind of an antibody will be having? He'll be having anti-A. You get my point? If a person's blood group is AB, he will not have anything. Why? Because he has both the type of antigens, right? He can recognize both A type of blood group, both B type of blood group. That is why his blood plasma will not have any type of antibodies. That's why AB blood group people are called as universal acceptors. Okay, universal acceptors. They can accept any group of bloods. They can accept the blood from A people, they can accept blood from B, they can accept blood from AB, or they can accept the blood from O people. Correct, right? Got it? You get my point? Now, come to O. If you look at O people, we understood that O people do not have any antigens. So you see here, there are no antigen in O blood group. But what you can see is in their blood plasma, they have both anti-A as well as anti-B antibodies. That means these people can donate blood. They are called as universal donors. They can see, as a O blood group person, I can donate blood to A, I can donate blood to B, I can donate blood to AB as well as O. But can I accept blood from any of these groups? No, I can't accept blood from A, I can't accept blood from B or AB. Why? Because my blood plasma has fighters. What fighters? Antibodies. What antibodies do I have? I have both anti-A as well as anti-B antibodies. So that's why it will avoid, okay, it will avoid me from accepting any other blood group except for O blood group. So O blood group people are called as universal donors. We can donate blood to anybody, and AB blood group people are called as universal acceptors. Okay, O blood group people are called as universal donors. That's correct, Aishwarya. Okay, so did you guys understand? So this is a very important concept for you guys to just simplify. So understand the difference between an antigen and an antibody. So when you hear the term called as antigen, you know that according to that blood group, you have that particular antigen. A blood group, A antigen. B blood group, B antigen. AB blood group, AB antigen. Then O blood group, no antigen. Whereas when they ask about antibody, it's opposite. A blood group, anti-B. B blood group, anti-A. AB blood group, no antibodies. Whereas AB blood group has both anti-A and anti-B antibodies. I hope this is clear. Yes, no, people? Okay, if you have any doubts, yeah, perfect. Okay, which blood group is known as best or effective? Now, this is a question, okay? Now, which blood group is known as best and effective. Now, the thing is, whenever you have a situation of surgery, whenever you have a situation of blood loss, then you need an immediate blood supply, correct? No, we can't go looking for, blood groups. So now at the donation state, whenever you want to donate blood, okay, then O blood group is perfect. Okay, you can save life by donating. If you want to save your life, okay, if I have to be saved, then I have to be an acceptor. I have to be an AB blood group. I can vouch for both O and AB. Okay, in order to save life, I can say that O blood group is very effective. In order to save myself, I want AB blood group because I can accept anybody's blood. So that will answer your question. So O and AB are efficient in their own ways. Okay, 50-50 I can vote. Okay, agree with me. Chalo. Now, before we answer this question, like if a parent is this kind of blood groups and parent 2 is this kind of blood groups, we have to talk about the type of alleles that are present in the particular blood group. Okay, so let us look at this. Okay, now let us say that we have three types of alleles. What are those? One is IA, other one is IB, other one is small I, correct? No, three types of alleles are responsible for the blood groups. Okay, I hope you can see the ink here. Yeah, yes or no? Can you confirm? Can you see the ink? Can you see IA, IB and small I? Can I write here? Make faster, faster. Can you confirm, anybody? Yes, okay, thank you. So now listen, okay, somebody says no. Okay, hold on. I will change the ink color. Let's go for black. Okay, now you tell me. Now, if I take about A blood group, okay, now also it's not visible. Hold on. Yeah, little bit now. Visible. Okay, fine. Now, if I talk about A blood group, so alleles, what allele should be there? Either they can be in a homozygous condition or they can be in a heterozygous condition, guys. For a blood group to be possible, it should be IA, IA or IA small I. Okay. The small I is an allele that is responsible for O blood group. You know that, right? It's a homozygous O blood group. So it does not play any role in any protein synthesis. It does not synthesize any polysaccharides or it does not express at all. That's why having one IA will always give you A blood group. Now let us talk about B blood group. B blood group is also same. Either you should have IB, IB in a homozygous condition or in heterozygous condition, your allele should be IB and small I. Okay. Next case is your AB blood group. In AB blood group, the allele should be IA and IB. You get my point? And then you have your O blood group. In the O blood group, it should always be in the homozygous recessive condition, that is small I and small I. So these are the allelic combinations. You know that for an allele to be expressive, they have to be in the form of pairs. So which of the following pairs will give rise to the particular blood groups? If I have to get a blood group, a person should have alleles either IA, IA or IAI, small I. Or if the B blood group should be there, IB, IB and IB and small I. And if the AB blood group is there, then I should have IA, IB. Then O blood group is there, then that means I should have small I and small I. Now listen to me. Now you can predict if my parent is having A blood group, and then if my other parent is having also an A blood group, what are the possible blood groups in the siblings? It's very easy. The table is very easy. They can be either A or they can be O. The same goes with A. If a parent is A and if a parent is B, what are the possible allelic, what are the possible children they can have? They can have children with either A or B or AB or O. Same, A and AB. Either they can be A or B or AB. If they are A and O, again, they can be either A or they can be O. Again, B and B, similarly, see here, A and A, AO, B and B, BO, B and AB, A, B and AB. So you just go through this, okay? You just go through this. Now, you can use it as an assignment to work out what are the possible ways. If a father or if a mother is A blood group, And if a see, they can give like this: if a mother is of A blood group and if a father is also of A blood group and the child is of O blood group, what will be the alleles of the parents? They will ask you this question. Then you should think, oh, what are the possibilities? Okay, if my a mother, okay, if a mother is A blood group, that means she should have IA and small I. Correct, no? And then if the father is also of A blood group, okay, let me take A blood group, IA and small I. Then what are the possibilities? See here, IA, I will get IA. So this will give me what? A blood group. Next one, IA, small I will give me what? Again, A blood group. Then again, IA, small I will give me what? A blood group. Then here, small I and small I will give me what? O blood group. Now, did I get my O blood group here? Now, did you understand how to frame a question with respect to blood grouping? Now, they can ask you anything question. If a parent is A blood group and if the other parent is AB blood group, or if one of the parent is having the blood group A and the daughter was born with a blood group AB, okay, what is the blood group of another parent? You understand my point? So you can work it out. You can just use it as a fun and try to make crosses and you will understand what are the possibilities. Either they have to be in the homozygous condition or they have to be in the heterozygous condition. If you make such crosses, you can solve any questions that come across. Last one is not visible. Which one? This one. That is small I, that would give us O. I just conducted a cross here. Okay. Okay. You understand, right? Are you following me, guys? Did you understand or you want me to do another cross here? Yes, no, can you confirm? Okay, table, last one. Last one is OOO. If a parent 1 is O blood group, parent 2 is a O blood group, then you have the possible child is also O blood group. Okay, that is the last one. O and O. Okay, so that is the last one. Clear? Chalo. Yes, before that is if a parent 1 is AB blood group and a parent 2 is O blood group, then the possible children will be either having A blood group or B blood group. Okay, parent 1 is AB blood group, parent 2 is O blood group, then the possibility is A or B blood group. Okay, yes, chalo. Now we understood this is how the blood grouping works. But did we counter our question of what is codominant condition? Among all the blood groups, what you can see is there is an expression of either one of the alleles in a heterozygous condition. So what did I talk about? Now, in a heterozygous condition means they should be of dissimilar alleles, correct, no children? They should be of what? Dissimilar alleles. Now, where all did we encounter dissimilar alleles? Let us look at this case. In this case, we encountered that we have IA and small I is a dissimilar allele. It's a heterozygous allele. What was the outcome? The phenotype was of A blood group. Again, you had IB and then you have small I. Here we had a B blood group. And the third case we saw was IA and IB. Here is also a heterozygous condition, but the phenotype was AB type of a blood group. Now, this becomes a matter of curiosity. In the incomplete dominance, we saw that there was an entirely different phenotype that came into an existence. But in a codominant situation, we are seeing a combined phenotypic effect. Correct, right? We are seeing a combined phenotypic effect. That is why through the blood group of AB, we can understand that whenever alleles are in heterozygous condition, both the alleles express equally of each other. Okay, so when they equally express each other on the plasma membrane of the red blood cells, you have both what? You have both, you can see here, you have both A and B antigens. When you have both A and B antigens, that makes your AB blood group an universal acceptor, and that's a beautiful example for codominance. Correct, no? Chalo. So codominance is also another example where it does not follow Mendelian law. Clear? So let's go for law of inheritance of two genes. Now Mendel worked out with a monohybrid cross. And in a monohybrid cross, we used one gene which one we chose one character, height as one character, and we took the alleles responsible for the height, and we understood few laws about it. We understood that they are in pairs. We understood that there can be a characteristics of dominance and recessive, correct, right? And we also understood that even though they are dominant and recessive together, during gametogenesis they segregate independently of each other. So this was about one character. But what happens? Mendel was curious. If I take two characters, okay, if I take two characters, or if I take two different alleles and two different characters, how would the genes behave? So for this, what he chooses is, he will choose the shape of a seed and the color of a seed as the example. So now this is a dihybrid cross, means there are two characteristics involved in it. We are very much aware of this cross because probably because of your CBS examinations, you would have worked out this Punnett square as thoroughly as possible, which is a very, very easy way to work out especially. Okay, I will show you that trick as well, because we should counter both the ways. Anyways, so what did he choose? He chose, one is the color of the seed. I'll write it here. One is the color, other one is the shape of who? Of the seed. Okay, which seed? Pea seed. So we know that the color is of two types. What are the colors? One is yellow, other one is green. So these are the color, correct? No? So these are the combinations. Yellow is dominant and the green is recessive. Similarly, we have the shape. The shape is round, other one is wrinkled. Okay, round is dominant. Okay, sorry, round is dominant, and then wrinkle is recessive. Okay, so that is why we take the dominant alphabets. Okay, so yellow is for Y, so we take it as capital Y. Capital Y are the alleles. Green is recessive, so we take small y, small y. Round is dominant, so that's why we take capital R, capital R. Wrinkled is recessive, that's why we take small r, small r. Now understand this point. Whenever we talk about one character, what we are talking, whenever we are taking in a dihybrid cross, we have to address both the characteristics together. So if I talk about a round and a yellow homozygous seed, a round yellow homozygous seed. So round yellow, you understand. Round is a shape, yellow is a color. Homozygous means what? They should be of similar alleles. So you know that if I talk about round and yellow homozygous seeds, that means they should have what? Their allele should be capital Y, and then you have capital R, capital R. Here we are using 4 alphabets. Remember, for each character, 2 alleles. That's why we have to represent all the 4 of them. Clear, no? Next one, we have the recessive type. If I tell green and wrinkled, then what will happen? Small y, small y, small r, small r. So this is the homozygous pure allele. That is, you have round and yellow, and then you have wrinkled and green. So these are the 2 parents we take. So when we take parents, what will happen here? How do we conduct the cross here? I will write it here. So first of all, in a dihybrid cross, we have the parents. So I will write it as P. The parents were capital R, capital R, capital Y, capital Y. You can vice versa also, you can write it. It's fine. And then you cross it with small r, small r, small y, small y. Correct? No, these are your parents. If they produce gametes, you know that they have to produce haploid gametes. Now parents are in diploid. Their gametes will be in haploid. So what will be the gametes, guys? I will represent it as G. So I will take out of R and R, I will take 1 R. Out of Y and Y, I will take 1 Y. And then they cross it with small r, small y. Now, if you ask me how, then you have missed a lot of class. Okay. Now, what will be the F1 generation, kids? F1 generation will be capital R, capital Y, small y. So it understands that all of them were round and yellow in the F1 generation, correct? No? So when he crossed with a yellow and a wrinkled, that is round and yellow and wrinkled and green, what will happen in the next generation? All of them will be round and yellow. Now the same technique he uses. What does he do? He will self-cross the F1. When he self-cross the F1, now what are his results? Now here is how you can conduct a, you can draw a Punnett square. How do we draw a Punnett square very effectively in a most easiest manner? First we take, look here guys, don't worry about the concept. Let's follow a simple trick. Okay, we need two parents. One is male, other one is female. Let's say that this is male and that is female. Okay. Now what do we do? Capital R, capital Y. Okay. Capital R, small y. Small r, capital Y. Small r, small y. You got it this side? Now replicate the same thing. Capital R, capital Y. Capital R, small y. Small r, capital Y. Small r, small y. That's it. That's it. Now you have the gametes of male and female. You can either way mention this is male, this is female, that is female, this is male. Either way it is correct. Now match this. R, R, Y, Y. Capital R, capital R, capital Y, small y. Again, capital R, capital small r, capital Y, capital Y. So like this, you start constructing. So case this R, this R, this Y, this Y. Like this, you keep on constructing. Finally, you will get around how many? 9 plus 3 is 12. 12 plus 3 is 15. 15 plus 1 is 16. Totally 16 different varieties you should get. Are you understanding my point? My idea is very simple. The thing is, many of the students, when you write in the board examinations, when you write your paper in the board examinations, what you do is you forget to take the combinations here. Some people will write RR like this. No, guys, no, don't do that. Okay, one to one combination. If I take R, I should go for Y. Go for that. If I take both capitals, one capital, one small, one capital, both smalls. That's it. Very simple, right? It's so simple. So you do the same thing and construct a beautiful square. If I construct a beautiful square, my F2 generation is done. So in F2, what I've done, I have self-crossed the F1. This plant, which one? R, R, Y, Y was crossed with R, R, Y, Y. So when I did that, what did I get? I get an F2 generation. If I look at my F2 generation, what do I see? I see that, can somebody tell me the genotypic ratio? Can somebody tell me the genotypic ratio of a dihybrid cross? It should take few seconds. Let me check. Not a phenotypic ratio. I'm specifically telling, can somebody tell me what is the genotypic ratio of a dihybrid cross? We have students from Andaman. Okay. We have students from, I can see some schools. Okay. So next time you just mention your school name as well. Okay. 1:2:1, 2:4:2, 1:2:1. Excellent answer. It's a very simple one. Okay. Whenever a genotypic ratio is asked into your dihybrid cross, remember that it is 1:1, 2:4:2, 1:1. Simple, no? 1:1, 2:4:2, 1:1. This is called as genotypic ratio. Sir, how? Guys, you have a lot of time. Please go through the square here. So RR, YY is only 1, so that will become 1. Similarly, there are only 2 types of some other things. Like that, you keep on grouping. Finally, you will get a genotypic ratio. Now you know what is a genotypic ratio. Whatever is the combination of these letters represent what? Alleles. So whichever have the same combinations, we put them in groups like 2, 2, 4, 2, 2. Whichever are unique, they come as 1, 1, 1, 1, 1. So if a genotypic ratio of a dihybrid cross will be 1:2:1, 2:4:2, 1:2:1. Very easy way to recognize and very easy way to remember this. Please remember 1:1, 2:4:2, 1:1. As simple as that. Agree? Chalo. Now what happened? Why did we conduct this cross? If I look at my cross, I get a very beautiful phenotypic ratio. Phenotype means what? When I look at my plants, I can recognize a specific physical characteristics. So what are the physical characteristics I came across? I understood that 9 of them, okay, sorry, it is a G. 9 of them were round and yellow. Round and yellow. Okay. Now, 3 of them were wrinkled and yellow. Wrinkle and yellow. Agree. And then again, 3 of them were round and green. Okay, round and green. My handwriting is bad. I'm sorry. Okay, 1 of them was wrinkled and green. Wrinkle and green. Now, this phenotypic ratio tells us very important thing. What is that? We have 9 of them were round and yellow, 3 of them were wrinkled and yellow, 3 of them were round and green. So, sir, where do we extract? Where do we understand a concept out of this cross? You have to observe these two. What are those? 3 of the seeds are having one recessive and one dominant character. Wrinkled is a recessive character. Correct? No, it's a shape. Wrinkled is a recessive shape. Yellow is a dominant color. Again, you see here, round is a, okay, round is a dominant shape. Green is a recessive color. Correct, right? So, in a dihybrid cross, what we can recognize is we can come to a conclusion of law of independent assortment. What is law of independent assortment? See, in my F1, when I self-pollinated my plants, they were having in a heterozygous character. Look here, there was one capital R, one small r, one capital R, one small y, one capital Y, one small y, like this. They were all in heterozygous condition. After I conducted the cross, okay, it did not matter whether they were going together with a dominant, whether they were going together with a recessive. Okay, so let us draw a definition here. In a heterozygous condition, okay, when two alleles coding for two different characteristics in a heterozygous condition during gametogenesis, they segregate independent of each other. Very simple, no? Means what is the meaning? The meaning is if I have two different characters, and if they, let us assume that these two characteristics are mixed with their dominant and their recessive alleles. So when they form their gametes, okay, we had what combination of gametes? A capital R gamete went with the capital Y allele, not gamete, allele. A capital R allele went with the capital Y allele. A capital R allele went with the small y allele. Again, a small r allele went with the capital Y allele. Similarly, small r went with the small y. So this is what is called as law of independent assortment. So whenever we have in a heterozygous condition coding for two different characteristics, the alleles segregated independently of each other. Okay, it did not control. They were present on the same locus of chromosome. That is another thing. The shape of the seed and the color of the seed, they were present on the same loci of the chromosome. But did the dominance of a shape control the recessiveness of a color? No. Did the dominance of a color control the recessiveness of a shape? No, they were all independent. That's why we got the variation. What kind of a variation? You can see here, wrinkled and yellow we got, round and green also we got. A combination of dominant and recessive was possible only because the alleles were left to independently assort during the gametogenesis. Did you guys understand this? Yes or no? Are you getting the concept? Sirisha, why Sirisha? Your ratio is correct, but the ratio you told is a phenotypic ratio. Okay? Is it clear, guys? Yes, no, fast, you should respond. Okay. I will wait for your response. Next one is chromosomal theory of inheritance. Okay. See what will happen as the time passed. As the time passed, people were getting more and more confused. People were not able to reject or accept Mendelian principles. Mendel was right, absolutely right. He statistically, mathematically, he's right. But somewhere, if I look at higher organisms, or some organisms, or let us call it higher organisms. If I take human beings, let's say we have a mother, we have a father, then we have a child, and the child is not looking like mother or father. Now, where did these parental characteristics go? What happened to these characteristics? So that was the biggest question in the history of biology. So what is happening to these characteristics? Okay, that was a problem. You get my point? So to answer this, okay, then comes your chromosomal theory of inheritance. Who proposed the chromosomal theory of inheritance? In match the following questions, you will get this. Okay, what is the contribution of certain scientists? So Walter Sutton and Theodore Boveri, okay, Theodore Boveri, they are the scientists who worked okay, on the chromosomal movements. Okay, they recognize that during meiosis, the chromosome behave in a certain pattern. Okay, so the certain pattern in such a way that you can see that in mitosis though, if there is 2n in the parental cell, in the daughter cells also it is 2n. Only in meiosis you can see that there is halving, or the alleles are becoming half in number, correct? No, 2n will give rise to n number of cells, okay, or haploid number of cells, or haploid cells, we can tell. So this behavior of chromosomes, that is from diploid to haploid, how they are becoming half, and then again fertilization, correct? No, see, for example, I have this is a mother, and let us say this is a father, and now they are, what is their ploidy? They are all 2n, correct, right? And they produce gametes. What is their gametes? Their gametes are n and n, correct? No, the sperm and ova, or whatever it is. And then what will happen? Fertilization happens. Fertilization leads to what? The formation of a zygote. Now the baby, is it haploid or diploid? It is diploid, correct, right? Now what does it contradict? How does it look like? Does it look like capital T going with another capital T and giving rise to TT, or capital T going with small t and giving rise to capital T small t, or small t going with small t and giving rise to small t small t? Like how we match the alleles during the monohybrid cross and the dihybrid cross, similar patterns are observed even in the chromosomes, right? Even chromosomes are exactly behaving like alleles. That's what these guys recognized during the meiosis. Okay, now, after they recognized, okay, so they understood one thing. Whatever Mendel is trying to refer it as factors, the same kind of behavior I can see even in the chromosomes. Okay, so that's why they gave something called as chromosomal theory of inheritance. So one of the best things these guys did was they never said that Mendel was wrong. They appreciated Mendel to the highest level. They said that Mendel is a genius. He told this without even having microscopy, without even knowing that there are something called as DNA, without even knowing that there is something called as chromosome. He was able to predict the pattern of how they segregate. So they appreciated his knowledge, and they said that Mendelian principle is absolutely right with respect to alleles. The same kind of behavior, how he explained about alleles, the same behavior we are also can see in the chromosomes. You understand my point? Chalo. So that's when the chromosomal theory of inheritance came. So what does it say? According to this, chromosomes and genes means alleles, no? Okay, they are all present in pairs, correct? No? They are all present in pairs. Now, in homologous chromosomes, what will happen? They separate during the gamete formation during meiosis. Same way how they separate, how the alleles separate during meiosis, chromosomes also separate during meiosis, correct, right? And fertilization afterwards, what will happen? The chromosomes which became haploid, they'll become what? Diploid. So this is what Walter Sutton and Theodore Boveri, actually these are the two individuals, okay? The great scientists, Walter Sutton and Theodore Boveri. Now what will happen? Thomas Morgan Hunt, along with students and colleagues, they started conducting the same experiments that Mendel's dihybrid cross. Okay, they tried to mimic the Mendelian dihybrid cross, and they wanted to see and analyze. Okay, they are telling that chromosomes also behave like alleles. Now, what is the correlationship between these two? Is there a difference or is there a similarity? They wanted to check, and they wanted to give reason where is this non-parental characteristics coming. Why exactly like parents the children are not born? Why there is new characteristics? That was his investigation. Who? Thomas Morgan Hunt. So Thomas Morgan Hunt, what he did was he worked on the Drosophila of genetics. Who is a Drosophila of genetics? It is Drosophila melanogaster. So Drosophila melanogaster, we also call it as Cinderella of genetics. So I call it Drosophila of genetics, sorry. Cinderella of genetics. Why Cinderella of genetics? She is having a very less number of chromosomes. She has a very small lifespan. Okay, you can see she has a very small he and she. Okay, they have a very small lifespan. They can produce enormous number of offsprings. They produce large amount of variations. Variations are important, right? If the variations are small, can you make a big data? Can you make a very solid data? No. Variations are large. Their lifespan is small. They produce a large number of offsprings, and they have a very short lifespan, and they can be grown in a very simple lab setup. Okay, they don't need very complicated lab setups. Within a very small lab setup, you can grow as many Drosophila as possible within a short period of time. So that's why you chose Drosophila. And moreover, the best thing is you can clearly differentiate between how does a male Drosophila differ from a female Drosophila. Okay, so that's why the Drosophila was chosen. Now, before we understand, how the Thomas Morgan Hunt's experimentation leads to understanding the chromosomal behavior in inheritance. Okay, there is another contribution. Okay, what is the time? Oh God, 4 minutes. Now the students, listen. Hold on, just a second. We have to continue in the next class anyways. Just be online. I want to give you some important information. The information is, just listen to me as